And I'm happy to invite and introduce the first speaker, Dr. Lynn Rothschild. She's a senior scientist at NASA Ames, and she's a bio-inspired technology lead for NASA headquarters. Among other academic titles, she's a faculty advisor of the Stanford Brown iGEM team on synthetic biology. And she has pioneered the use of synthetic biology for space exploration, astrobiology, and even Mars settlements. And in 2015, she was awarded the Isaac Asimov Award, which is, I don't know for what exactly, maybe for novel or for what? So maybe she can tell us. And she's been many times a NIAC fellow for NASA as well. So her title of her work is Universal Biology and Life How It Must Be. Please.
Thank you. And the answer is, it is a great mystery to me how I got the Isaac Asimov Award, but I can tell those of you who are aspiring to it that you don't actually have to have read any Isaac Asimov or having written a novel to get this award. It just is some great mystery. Ah, great, there we go. So what I'd like to do is ask the question about how much you can have biology treated like it's physics. So here's a picture I took in Westminster Abbey. You can see Darwin's stone there, Newton's around the corner, and Newton's there, I imagine, after hours asking Darwin, you know, is biology like physics? Can you write equations, F equals ma? And he said, well, you know, I'm not entirely sure about it, but there could be, you know, aspects of it. And so that's what I'm going to address today. Stephen Hawking, I check, has not been interred there yet. He'll be somewhere in that area, so we do not need to bring him into the conversation until the next lecture. OK, so this is a question, could you do it again? I used to call this talk replaying the tape, but as the new generations come through that doesn't understand what tapes are, we have to change the title. So the point is that this is extremely important if you're going to ask the question, are we alone? And I always like to say, we have a problem that we only have one data point for life. And so it's like me going into the English department at Stanford and asking for a job. And they say, well, how many books have you read? And I say, one. I would be laughed out of the room. But that's the position that we're in as astrobiologists. We only have one book of life. And it would be incredibly naive of us to say, this is the only way it can be done. Now, it would be actually incredibly interesting if it turns out there was only one way to skin a cat. But I suspect that that's not true. And so what I'm going to try to do in this talk is to give you an idea of how we might go about this, knowing that we only have one example of life. So today's points are going to be asking what actually is life. Obviously, there's no really good definition, as the introduction indicated. But how we can argue, and that's from things like physical constraints and from convergence, which gives us some kind of pseudo-replicates, and then some constraints that the environment gives on us. This great picture of a puppy is there not because it has anything to do with the talk, but I couldn't think of anything else, and everyone always says aw when they see a puppy. So you're supposed to say aw every time you see the puppy. Thank you. So the question is, what is life? And Carol Cleland brought this quote to my attention a few years ago. It is absolutely brilliant. This is Da Vinci defining life. Water is water. Sometimes it's sharp, sometimes it's strong, sometimes it's acid, sometimes it's bitter, sometimes it's sweet, and this goes on and on. That is, of course, not how we would define water today. We would define water with a natural definition. Water is H2O. And I think this pretty much sums up the question of how you define life. Is there a Da Vinci sort of definition only, which is what we're talking about now? Does it look like this? Does it undergo Darwinian evolution? Is it made of that? You know, it's sort of like the blind man with the elephant. Or is there some kind of fundamental natural definition, an H2O definition? And to really show my cards, I don't necessarily believe that there is an H2O definition for life, and that in the end, it may be a matter of taste. So if we go with the Schrodinger, it has to fight entropy, and someone says, well, what about a flame? It may technically fit that definition. I don't feel comfortable calling a flame alive. And so it may be that in the end, like I say, there is no real essential natural definition. And of course, the conclusion is if that's true, that there could be an alien somewhere else that does not include us in their definition of life. Thank you. But we do need to, that reminds me which slide's coming up. So what can we do? We do need some kind of operational definition. I can't very well go back to my lords and masters at NASA and say, well, there is no natural definition for life, so you don't know what you're looking for. So one thing I think we can say is it's an emergent property that bubbles up from chemistry, and it's based on physical components, but it's a process rather than just an object. Otherwise, we wouldn't be able to distinguish between living and dead organisms. So this is critical. Life is a process that has some sort of body to it. So there's a corporeal component. I do think Schrodinger's point about fighting entropy is very important. Of course, looking around the room, I don't think there are any children present. That's why we all had lunch today, to fight entropy. I think Steve Benner and I both realized we weren't going to talk about chirality. There's the word chirality. And if anyone wants to talk more about that later, you're welcome to. Steve mentioned that a little bit in his panel comments. I believe that they're going to be physical components, and I'm going to make a big case for organic carbon chemistry. I believe Lee Cronin will be coming in later saying that that's total bunk, but the beauty is his plane hasn't landed yet. And then you need a solvent, likely water. Must it undergo Darwinian evolution? And Darwinian evolution is a great idea. Not everything on Earth is a result of Darwinian evolution. I look around the room. I know maybe 50% of people in the room, maybe 40%. I don't know if the rest of you were made in some chop shop where they've got all the construction going on by the Galvez parking lot. I'm going to give you credit for being alive, whether I see any Darwinian evolution going on or not. So let's look at the physical component aspect for just a moment here. Why carbon? Why is it a carbon-based compound? And I'm very, very bullish on carbon. Here is an abbreviated form of the periodic table. I'm a biologist, but here's an astronomer's periodic table, which is even more brilliant. And what you see here immediately is that there's vastly more hydrogen in the universe than anything else, than helium, which really isn't very good for life except for birthday parties. And then you've got oxygen and then carbon, which are big ones. And what's interesting is you notice a lot of these others, with the exception of argon and neon, that are high up there are also ones very important for life. So we see that there's lots of carbon available. There are lots and lots of very intricate compounds that can be formed by carbon. This is, I feel, let me stop for a second and say, I feel a little bit like Darwin, who was forced into publishing The Origin of Species, and he called it as an abstract at 700 some pages. And I feel like I'm trying to present an argument to you that I'm giving you a very tiny abstract. So you will be able to pick holes in this because I can't give you all the supporting data. I'm just going to give you the outlines here. So it is capable, back to the main show, of forming these complex compounds, and even better, our colleagues in astrochemistry tell us that they find this kind of carbon chemistry going on out in the interstellar medium. So it's not just a great idea, it's actually happening out there. We see evidence from meteorites and so on. If you have followed me so far, you already can make all sorts of hypotheses about life. Virtually every organic carbon compound, I guess all of them, absorb in the ultraviolet portion of the spectrum. And so UV radiation is going to be hazardous to every life form if every life form is based on organic carbon. Similarly, if you start to go up too much in the infrared, you don't have enough energy to run life. And so you're starting to constrain yourself into these wavelengths of about 400 to 750 nanometers, which is where life on Earth plays. It's where we use our vision. It's where photosynthesis occurs, and so on. So you're already putting constraints on life just by saying organic carbon is involved. As I mentioned, a lot of the compounds, a lot of the elements that we believe are really important for life are some of these ones that are really abundant. Maybe they've been chosen for the reactivity. Maybe it's a combination of reactivity and abundance. Water, I'm not quite as bullish on water as a solvent, although people like Steve Benner, who I admire enormously, I know has worked very hard in being perverse in finding other sorts of solvents, and I think that even in the end, and he can correct me if I'm wrong, that water does turn out to be a pretty darn good one. There are all sorts of reasons for that have to do with its chemistry. It is also wildly abundant in the universe, back to the astronomer's periodic table. So, thank you. So we have this point. If you start with the organic carbon chemistry, if you add water in, we're already making hypotheses that would be universal for life. I'm going to assume that the laws of chemistry and physics pretty much go across our universe. And finally, that the environment creates some kind of further constraints. So, for example, temperature. If you're based on organic carbon, you can't live above 500 degrees C because organic carbon is just simply not stable there. And even within that, we can start to put constraints in with pH and so on, because that's going to constrain your chemistry. Now, we have then the ability to go from the elements to start to look at things like amino acids. And what you see in this table on the left is part of a table that I admire enormously from a paper of Ralph Bedritz's. This is right there in the middle of the audience, showing the rank order from abiotic settings of the 20 or so amino acids that are used in ribosome-produced proteins in life on Earth. And what you see on the right are amino acid rank order of ones that are found from things like meteorites. And it's not too bad on that top 10 in terms of similarities. What you don't see is the chirality on the left. But you ask the question then, do we use these amino acids because they're very abundant? And I think the answer is probably yes. But when you start to play those games, you can start to say, yeah, I could see my way to amino acids being used in these life forms. They're out there. What about the folding? You can certainly polymerize amino acids abiotically. You can get impact synthesis and get polypeptides and so on. What about folds? The folding of these proteins can be done in many different ways, but not all of this phase space is used by life. Is it because there's been selection, or you can't do every one? Now, what's interesting is I found a paper relatively recently, showing that the beta sheets, the ones that are formed in amyloids, similar to what's formed by prions, you get the amyloid plaques for Alzheimer's and so on. This beta sheet conformation has been proposed to be actually one of the earliest. It can be done with a very tiny peptide. It's very common, and it's got all sorts of resistance to the physical environment and so on. And so you can see where this is leading is, yeah, if you're going to get amino acids, you can get the peptides. If you're going to get the peptides, it's not unreasonable to imagine you would have beta sheets. Could you then turn these into polymers? Because many people talk about looking for polymers for life. As I mentioned, there's certainly shock synthesis and other sorts of syntheses of peptides. Certainly people like Dave Deamer have shown that you can do abiotic formation of RNA, so you can polymerize the ribonucleotides abiotically. So again, if you've got those components there, there are all sorts of ways for this to happen. DNA, not so easy. So in conclusion to the first part of the talk, if we replayed the tape, I do firmly believe that we would be organic chemists again, probably using liquid water as a solvent, and this would suddenly put all sorts of constraints on us. And so the sort of the conclusion is, if there's life around Alpha Centauri, they're using roughly the same biochemistry books that we use with life on Earth, except obviously not in English. So the conclusion is, don't look for aliens that are too exotic. And it's not because I don't have an imagination, it's because if you start to follow these arguments, what the world's actually telling us, there's not an infinite number of ways that you can do this. So the second approach that you can use is to argue from convergence. As I mentioned, we only have this one example of life. So I think someone mentioned earlier in the panel that, oh, it takes 1 billion years before you get a multicellular life form, or you get this or that. We have one book of life. It doesn't mean that that is always the way it's going to happen. And by the way, I know everyone in this room throws around numbers like 1 million years. If I say it takes 1 million years for life to form, you say, oh, that's an instant in geological time. But just stop for a moment. That's a really long period of time. As humans, we think in 100-year time scales. Even 1 million years would be a lot of time. I can't promise you that life couldn't arise in 1 million years. You can do all sorts of calculations, but it doesn't prove that you need to have exactly the conditions and exactly the trajectory that happened on Earth. So let's look at this argument from convergence. And again, I realize that philosophically these are pseudo-replicates because we're starting with life on Earth, but it's better than nothing because we only have this one book. And this is a very simple sort of argument. You see these pictures here, the cactus, and a rose there, and a whistling thorn acacia. Of course, that top middle one is the rear end of a porcupine. And every one of these pictures shows the same thing. These are independent solutions to the problem of being eaten. So it's basically saying, do not bite, do not eat me, it will hurt you. all evolved completely independently. And so it gives you the feeling that if you have a certain level of life, whatever the common ancestor was for the organisms represented in these 4 pictures, that you have the capability to come up with the same solution to that problem. So let's look at a couple of these, what I call great moments in evolution, and see whether those are likely to happen again by convergence. So let's look at metabolic pathways. I've discussed a little bit about that earlier, the peptide synthesis and the folds and so on. But let's look at one of the really big ones, the evolution of photosynthesis. I talked about that in a paper a few years ago. And the conclusion is that the ingredients for photosynthesis, radiation, carbon dioxide, the ability to use a reductant, are very widely available, multiple places, probably every place that you would be looking for life. That we see convergence going on over and over in the metabolic infrastructure that's needed. So the evolution of carbon fixation enzymes occurred over and over and over again. It wasn't just once. We have light receptors that arose independently, the ability to capture light energy and turn it into chemical energy. We have these different components that have arisen convergently, plus there was the evolutionary pressure. That with Titan aside, there's a much better chance from what we know about other places in our solar system to find inorganic carbon than organic carbon. And if life is based on organic carbon, at some point, you're going to be forced into making your own if it becomes a very limiting feature. And so any organism that can figure out how to make their own carbon and use light in particular, but there are other sources of energy too, you're going to be a huge advantage. So the argument is that that could, I believe, happen again. What about multicellularity? I think I heard some snide remarks in the panel earlier about simple life. Put on this table, there is no such thing as simple life. The gap between non-living and living is the huge gap, not as many people in the public think, between a microbe and a human. The gap is between non-living and living. Life was complicated, and don't feel too arrogant about being multicellular. If you look at the domains of life, we're there at that very little tip. But if you look at a lot of those other organisms, they are what I call temporally multicellular. They have different life forms. They have different cellular types at different stages in their life. These 3 groups arose independently. There are lots of great reasons, lots of evolutionary pressure for multicellularity, avoiding prey. Diffusion problems if you stay unicellular, but you try to get big. You have problems with senescence, sequestering the germline, and so on. What about the transition to land? That happened over and over again. Again, the evolutionary pressure for that is clear, that you have a much higher concentration of gases, particularly if you're in a high temperature. The solubility of gases goes down as the temperature goes up, and so a great way to avoid the fact that you're starving for oxygen or CO2 or whatever you get off on is to go out into land. Now, of course, you then have to face desiccation and so on, so there are problems with that, but obviously organisms have solved that problem over and over and over again, and so that gives you some confidence. What about intelligence, speaking about being arrogant in the room? That even amongst the mammals occurred over and over again in several completely independent lineages. We could certainly make an argument that the cephalopods, things like squid and octopus, are intelligent as well. Again, would be a different origin, but I don't want to get off into the SETI track. I did want to finish with a sort of following up on the comment. Chris said that I was part of the synthetic biology mafia. Yes, but even before that, I was part of the protistan mafia. But we'll get back to the synthetic biology mafia, and I completely agree with Steve's assessment that the first place we find an independent origin of life is going to be in one of our labs. And so using a laboratory approach, I think, is going to be extremely fruitful and much quicker than many of these others. So let me just give you a quick example of something that has been done, and that's looking at this issue of the evolution of multicellularity. And this is work of Will Radcliffe when he was working on his PhD at the University of Minnesota. And they did something very simple-minded, but in the nicest possible way. I didn't mean that as an insult. I'm a huge fan of very simple-minded experiments. Here they took Saccharomyces cerevisiae, which is baker's yeast. It is unicellular, it's a budding yeast. And they asked, could they make it multicellular? And here's the simplicity to this. They shook it up, They let them sink in a test tube and sat there with a stopwatch, and the ones that sunk the fastest, they then moved into a new test tube. And so they did this selection passage system about 60 times, and within 60 passages, they got what they call snowflake phenotypes. By the way, I didn't mention Mike Travisano was the PI on this. And here's some of those snowflake phenotypes that they got, and these then became heritable. So it was within 60 passages, you had the evolution of multicellularity. Now, in fairness, there is some thought that yeast were originally multicellular and they're secondarily reduced. So they have started to try this sort of thing, for example, with Chlamydomonas. But what's interesting is that you can repeat that, as it sort of emphasizes my point that the evolution of multicellularity is fairly simple in the grand scheme of things. What about making life de novo? Lots of people working on it. We're at the point that we even have students in the lab working on this sort of thing. You certainly can do transcription translation systems. You can encapsulate them into membranes of various kinds. The flagellum's just there for decor. So back to Westminster Abbey. Here we've got, again, Newton asking Darwin, is there something about life that allows predictability? Are there any laws? And I think the answer is sometimes yes, sometimes no, but if you even start by this idea that life is based on organic carbon chemistry and you add in that water is a solvent, you can start hypothesizing all sorts of things and go from there. And so that does actually constrain our search space for life elsewhere to some extent. It doesn't completely constrain it. It doesn't mean that I'm going to look for a green-eyed, mirror image of myself somewhere else, but we can at least get some idea and therefore help design our instrumentation, so on around it. Thank you very much. One question? I was told time for one very good question, not to put the pressure on you. Oh, good, another member of the Proteastin Mafia.
Ramon Saranascos, Big Low Lamb. I have a question about synthetic biology. When we think about strategies to find life or to think how life may look like in other places, we look at Earth and we think about the history of Earth backward. If we think forward, things may be quite different. If we perceive with technological advances like synthetic biology and artificial intelligence, in another few thousand years or a million years, which is still a blink of eye in geological terms, we may have completely different forms of life and intelligence. So with that in mind, what can we think? Can we improve our ways to search for both intelligent and non-intelligent life?
Yes, and actually you're sort of catching me on that because I'm completely ridiculously overdue on a manuscript and the editor's sitting right near you discussing that very point. And I will cut to the chase, assuming that everyone here is completely honest about this, is that we're talking very much about biosignatures of meat, like us, protein-based life forms. And so we're looking for things like disequilibria and water and things like that. But if you have a future technology-based form that's based on, say, silicon, you would not want to be on a water-based planet because that would be very corrosive. You might want some place that's going to give you the energy to power you up for your silicon-based life form. So I think that you're right, that we would be looking for something very different. However, I did want to unpack a little bit of your comment that it's not in the future that we're adding synthetic biology or a human influence onto evolution. We've been doing that for 10 to 15,000 years. I tell people, you don't give a wolf to your kid for Christmas, you give them a puppy. You don't barbecue teosinte, you barbecue corn. We've already been dealing with changing the genomes of the organisms around us. We've been calling it domestication. We're adding this synthetic biology in, which is much more targeted, but we've already been affecting the trajectory of the evolution of life on Earth for, as I say, about 10 to 15,000 years. So it's already happening.